All right, guys. So one thing I forgot to mention last time is in these slideshows, I try to do like the most important slides thing because I sometimes have way too many, like more than I intended to put into the slideshow. So the number of the most important slides are here, and then I also put like an asterisk in the upper left corner of the important ones. So yeah, you can pay attention to that if you want to. Okay, last time I sort of left off talking about neutrophils. That was the last thing I talked about. And I told you that neutrophils are blood cells, right? They're in the blood. They have to be called into the tissue, recruited into the tissue by macrophages. And macrophages make a cytokine called IL-8 or interleukin 8 to recruit neutrophils into the inflamed tissue. And neutrophils don't live for very long before dying, but their job is basically to phagocytose things and kill them. Okay. And they're going to accumulate early, and later on, you're not going to see as many of them during the infection. They are a rapid responder, so to speak. Monocytes, on the other hand, they're a little bit more involved. So they are the second wave of cells that are recruited into a tissue. So they come after the neutrophils. And that's because they're recruited using a different cytokine. They are phagocytes. They can do a lot of cool stuff that neutrophils are not capable of doing. And this makes them a little bit of a more mature defender than neutrophils. So normally they're patrolling the bloodstream, looking out for bloodborne pathogens, and occasionally they leave the blood. Even when there's not inflammation, they will leave the blood and enter tissues. And the reason that they do that is because they differentiate into macrophages and they differentiate into dendritic cells. They can turn into either of those two cell types. So this is where some of your macrophages come from. Uh, Macrophages do have a limited ability to self-renew in the tissue, but monocytes also replenish them periodically at a low level. However, during inflammation, you're going to get the recruitment of a lot more monocytes. Okay, and they can present antigen to CD4 positive T cells or T lymphocytes using MHC2 and B7. Okay, so they can present exogenous peptides to T cells, and they are the only cell in the bloodstream that is capable of presenting to T cells, to T helper cells. And if they leave the blood to enter the tissues by traversing the endothelium, the recruitment of monocytes is not through interleukin 8, it is through CCL2, a different chemokine that isn't produced until later in the immune response. Okay, CCL2 doesn't come until the macrophage is stimulated with a pretty good dose of cytokines, usually in an autocrine fashion. So more stuff has to happen before the recruitment of monocytes can occur. They produce chemokines, they produce cytokines, they participate in ADCC, and they are the biggest cells that are in your bloodstream under the microscope. They're even larger than lymphocytes. They have a non-segmented nucleus, unlike neutrophils. Okay, so that's what monocytes do. They're really important to give rise to macrophages and dendritic cells. A bit of a different cell group is the granulocytes, and I put this here because I didn't know where else to put it, I guess. Now, neutrophils are a type of granulocyte, and I sort of mentioned that. But basically what a granulocyte is, it's just an innate immune cell that contains a cytoplasmic granule. And the reason we differentiate between these cell types is there's a few reasons. First of all, they have a different microscopic appearance, and they come from a different precursor cell than monocytes, macrophages, and dendritic cells. So they have a bit of a different lineage. The granulocytes are a group of 4 cells. They are the basophils, the eosinophils, the mast cells, and the neutrophils. All of these are granulocytes. They are not professional antigen presenting cells. Okay, they don't express MHC2, they don't express B7. They cannot present antigen to T helper cells, which are CD4 positive T cells. However, they are mildly phagocytic cells, especially neutrophils. They are very phagocytic. So they are capable of killing things intracellularly. But what you'll find is that basophils, eosinophils, and mast cells have a strong preference to kill things outside the cell. Okay, so mast cells, basophils, and eosinophils are sort of like a subdivision of granulocytes, like a triad of cells. They aren't very phagocytic, and they're very involved in similar things. Neutrophils kind of do their own thing in this group, but these three cells are very closely related in their activities. So they defend against extracellular eukaryotic parasites, things like worms and blood parasites and tissue protozoans and all those things. And they also, unfortunately, carry the side effect of being one of the main perpetrators of allergic responses to allergens. Okay? And allergens are just an antigen that is accidentally recognized as bad or accidentally triggers an allergic response. Okay? It's the body thinks that there's like a parasite there, but there's not. It's sad. And allergens are generally molecules that bind to IgE isotype antibodies. We'll talk more about antibody isotypes later on. So the reason that IgE matters is that IgE arms mast cells and basophils. So mast cells and basophils become armed when the Fc region of IgE molecules binds to receptors on the mast cell basophil surface called the Fc epsilon R1 receptor. This is also called the high affinity IgE receptor. And that's probably a more common name for it. So this is unusual. Usually antibodies bind to their antigen first, and then an immune cell can come and recognize that there's a bound antibody sticking to something. IgE is the opposite though. IgE first binds to its receptor before it binds to an antigen. So the sequence of events of binding is opposite for IgE compared to the other classes of antibody. Talk more about it later. So mast cells and basophils become armed when they bind to IgE molecules that have not yet bound to antigen. And then mast cells and basophils degranulate when the FAB region of the IgE then binds its antigen. So only then is the cell triggered. The cell already binds to antibodies, but those antibodies haven't bound to anything else. Once they do bind to their antigen, that triggers the degranulation, the activation of the mast cell slash basophil. Okay. And one of the most important substances in the immune response, because it's so rapid, is this molecule called histamine. So mast cell and basophils both make histamine, and they pre-make it before ever being activated. They store it in granules in their cytoplasm. And when their IgE molecules on their surface bind to an antigen, they release all that stored histamine, which triggers a really strong, really rapid inflammatory response. And this is part of why allergies can be so rapidly bad. Okay. Mast cells are present in the tissues. Even in the absence of infection, they hang out in the tissues and they self-replicate and they're sort of self-sufficient. And they carry out surveillance, so they're kind of like macrophages. Basophils and eosinophils, they circulate in the blood. That's usually where they hang out. But they can be recruited to tissues just like neutrophils and just like monocytes. Okay. So what does histamine actually do? How does it cause inflammation? So this is sort of an expansion on how inflammation can occur. This is true for histamine, but it's also true for a lot of the other inflammatory mediators. So histamine binds to its receptor, and it may not be histamine. You might be looking at tumor necrosis factor or some other inflammatory mediator. For histamine, it's binding to the H1 receptor for histamine on muscle cells that are located outside of local blood vessels feeding the inflamed tissue, okay, or the arterioles that are feeding the inflamed tissue. And when histamine binds to its receptor on smooth muscle cells surrounding vasculature, it causes those smooth muscle cells to relax. This results in vasodilation. The blood vessel gets larger, wider, and has a higher diameter. Okay. Vasodilation is important for two reasons. First, it increases the volume of blood reaching the inflamed tissue, and it increases the supply of blood to the inflamed tissue. And therefore, you also get more lymphatic drainage from the inflamed tissue. Okay, so your lymphatics begin to drain a lot more fluid because the tissue is taking up more fluid from the blood. Second, vasodilation is important because it slows the velocity of the flow of blood in that area, which means that immune cells aren't being whisked away by the powerful blood current in that area. Instead, they can sort of take their sweet time passing by all those vascular cells, and that allows them to sort of survey all of the molecules on the vascular cell surface. A lot of the important molecules on the vascular cells is going to be adhesion molecules. So when you slow the velocity of blood flow in the inflamed area, you have prolonged cell-cell interactions between the bloodstream leukocytes and the vascular endothelium. Okay, this allows blood cells to more easily be able to pull themselves out of the bloodstream and into the tissue where they need to fight the infection. So that's what it does to smooth muscle cells. That causes vasodilation. Histamine also acts on endothelial cells directly, and it's the same receptor, it just causes a different cellular response because it's a different cell type. So it binds to H1 receptors on nearby vascular endothelial cells, and it causes this really important thing called endothelium activation. And this again is two components, it's twofold. First, endothelium activation means that the size of the gaps between adjacent endothelial cells is increased. Because the endothelial cells physically contract and become smaller, they move apart from each other. They're still linked by desmosomes, okay, but those desmosomes are a little bit more lenient in terms of what can pass through them. This results in increased fluid and blood protein leakage into the tissue. So you have an increased volume of liquid getting into the tissue, which helps to drain everything toward a lymph node where it can be surveyed by lymphocytes. And you also have all of the proteins in the blood being leaked into the tissue, okay? And those blood proteins include things that can limit the replication of pathogens and mark them for destruction. Antibodies. You're also talking things like vitamin B12 binding protein, which binds to vitamin B12 and prevents it from bacterial uptake. It's an important growth factor for a lot of bacteria. And transferrin and lactoferrin. Those things bind to iron and sequester iron from bacteria so they can't use it for their growth. All of those things are in the blood, hence endothelium activation and increased vascular permeability, though that is an important thing for the defense. Second, endothelium activation does not only include increased vascular permeability, it also involves the upregulation of adhesion molecules on the endothelial cells. And those adhesion molecules are used by the circulating white blood cells to leave the blood and enter the tissue. Okay? So hopefully this clarifies a little bit about what's going on during acute inflammation. It's written out a little bit nicer here. And here is a couple of the things that eosinophils contain in their, well, this is just how eosinophils can kill, essentially. They can do phagocytosis to some degree. They're not very good at it, but they can. They also can release some of the stored toxic things in their granules, like major basic protein, etc. Sorry, my phone always falls during SI. They release toxic oxygen metabolites, reactive oxygen species, including oxygen, superoxide anion, hydrogen peroxide, etc., and a number of other things. They release lipid metabolites, eicosanoids, which cause inflammation, and they release cytokines. Some of those cytokines are pre-made, but a lot of them are made from scratch when the cell becomes activated. Okay, most cytokines are not pre-synthesized. This figure is from this really cool encyclopedia of immunology that I found. And then here's another table from, I don't know, I think this one came from like a Nature article, but you can read this on your own time. Basically, they kill things and they cause inflammation when they are activated. You don't like need to know these specific items. So this is what a mast cell looks like under the microscope, and you can see it here as well. They tend to stain pretty they tend to stain pretty pink when you use your H&E stain. So mast cells are in the tissues, eosinophils, basophils, and neutrophils are in the blood, and they are to some degree in the tissues when there is no infection. Okay, here's how the degranulation process works for your mast cells and your basophils. Okay, so antibodies, these little Y-shaped proteins here, Before they ever encounter antigen, the IgE class binds to a receptor for IgE on the mast cell surface like this. And this arms the mast cell against, a potential offender. And then it is only then that the antigen binds to IgE, and that triggers the degranulation of the mast cell. Okay, so this figure just shows you what I was trying to explain a second ago. And then we have natural killer cells. And these are lymphocytes, but they are innate immune cells. So unlike the other lymphocytes, which are adaptive, natural killer cells and the related cell types, they are innate cells. So natural killer cells are actually considered a class of what we call innate lymphoid cells, which I'll talk about in a second. They are not professional antigen presenting cells, and they are not phagocytes. They don't ever phagocytose things. Instead, what they do is they walk up to cells and they shoot them in the face. So they directly kill cells. Basically, natural killer cells are super important for cell-mediated immunity, which is a concept that I'll elaborate on later. So natural killer cells are activated by macrophage-derived IL-12 and tumor necrosis factor to a lesser extent. So macrophages activated by pathogens make a lot of IL-12 and they make a lot of tumor necrosis factor. Those cytokines act on natural killer cells. That's one of the targets of those cytokines, and that causes natural killer cells to get all pissed off. And they begin making a bunch of this cytokine called interferon gamma, IFN gamma, in response to that. And interferon gamma then acts on the macrophage and activates it. Specifically, what it does to macrophages, dendritic cells, and monocytes, it will increase reactive oxygen species production. It will increase the ability of the cell to fuse the phagosome with the lysosome, and that can be helpful to overcome some types of inhibition of the fusion that you see with certain pathogens. And it upregulates MHC2 and B7 on the surface of the cell, making them better presenters of antigen. Okay, so they become better at receiving T cell help as well. It also upregulates the production of IL-12, TNF, and other inflammatory cytokines. So you can see that this acts as a positive feedback loop. The IL-12 acts on the natural killer cell, causes it to make interferon gamma. The interferon gamma acts on the macrophage, causes it to make more IL-12. And they're just continually activating each other like clockwork in a positive feedback loop. Natural killer cells are not only interesting for their cytokine production activity, but they're also important for the production of cytotoxic proteins. And these cytotoxic proteins are pretty wide-ranging, but for the most part, they're going to be killing infected host cells. However, they do have an ability to kill some parasites and some bacteria and fungi, etc. Mostly they're targeting virally infected host cells or intracellularly infected host cells. Three of the most important things that natural killer cells use to kill. These are the most important ones because another cell type uses all three of these as well. Perforin is the first. Perforin is a protein that inserts into membranes and polymerizes into a pore. It literally opens a hole in cell membranes. And this is bad for obvious reasons. You get ion gradients being disrupted, and the cell is going to die because of that. Calcium is going to be leaking into the cell, which can help promote apoptosis, etc. But also, perforin is an issue for the target cell because proteins can be injected through that pore, and those proteins can be a battle all in themselves. Okay. One of the proteins that can be injected through perforin is granzymes, which is actually a class of proteins. One of the more important ones is granzyme B. Granzymes are actually serine proteases that cleave proteins that induce apoptosis. Okay, so they're going to trigger apoptosis through the intrinsic pathway. And then you have granulysin, which is an antimicrobial protein that can also induce apoptosis through a distinct mechanism from granzymes. Okay, so both of these are going to kill the cell. And for the most part, they're going to be entering through perforin. Natural killer cells are really good at killing targets. If you make a natural killer cell upset with you, you're going to regret it. There's virtually no cell type that is resistant to natural killer cell anger and destroying activity. Natural killer cells express this class of receptor called the killing inhibitory receptor, the KIR. So you may be asking, okay, these cells are really ferocious, they're really furious, and they can kill anything they want to. When do they know not to kill? They're just killing machines. When do they know not to kill? The KIRs, the killer cell inhibitory receptor, or the killing inhibitory receptor, is a class of proteins that tells natural killer cells not to kill. Okay? So the KIRs are expressed on natural killer cells, and they are actually a receptor for MHC1. Regardless of what the MHC1 what peptide that MHC1 molecule is holding, it's going to recognize MHC1. Okay, so it's not antigen specific, it's just a receptor for MHC1. And when KIRs bind to MHC1, they prevent the natural killer cell from getting upset. Okay, it's kind of like an antidepressant something. So viruses may downregulate the expression of MHC1 on infected cells. And that is a thing that they do because it allows them to evade detection by T cells. However, if a virus was to downregulate MHC1, that means it now has to deal with natural killer cells. Even though it's safe from T cells, the cytotoxic T cells, it's going to have to deal with the natural killer cells instead, because natural killer cells are inhibited by MHC1, and if the cell isn't expressing MHC1, the natural killer cell is going to kill it. If you don't have MHC1 on your surface, you very clearly have a problem. So the natural killer cell knows to kill you if you don't have MHC1 on your surface. So it's kind of like a double negative situation. It can be kind of hard to understand. There's a figure on the next slide that might help. Natural killer cell killing is also upregulated or enhanced by type 1 interferon cytokines like interferon alpha and interferon beta, which are cytokines made by epithelial cells, etc., during viral infection. And we'll talk more about those in the next slideshow. And natural killer cells are one of the most important classes to participate in this wonderful activity called antibody-dependent cellular cytotoxicity, which is abbreviated ADCC. Basically, this is where an antibody binds to a cell, and that cell is then killed by something that recognizes the antibody being there. Okay? Natural killer cells usually respond to antibodies of the class IgG, and they're going to secrete these perforin, granzymes, and granulysin in order to execute the targets of ADCC. Okay, and I have a figure on that coming up. So, two scenarios. First scenario, the inhibitory receptor is engaged. The natural killer cell expresses activating receptors and inhibitory receptors. One of the more important inhibitory receptors is the KIR, the killer cell inhibitory receptor. And all that it does is recognize MHC1 on the surface of the target cell. And when that happens, when it recognizes MHC1, it's going to send a strong do not kill signal to the natural killer cell. And the natural killer cell is going to say, okay, I'll move on with my day, and I'm not going to kill you. Okay, so the natural killer cell flies away, and it moves on to the next cell. So the natural killer cell does not get activated, and it does not kill the target cell if it's expressing MHC1 on its surface. On the other hand, if a virus is replicating in a target cell, and that virus downregulates the expression of MHC1 such that it's not really present on the surface too much, then the natural killer cell doesn't get an inhibitory signal. So it's not going to have basically a calm down signal. And in addition, usually if the target cell is infected with a virus, it's also going to be expressing a lot of things on its surface that indicate that it's in distress. So it's going to be putting danger signal proteins out on its surface that the natural killer cell recognizes, and that also tells it to kill. So not only do we have a missing do not kill switch, we have a present do kill switch. And so the natural killer cell says, oh man, the message is just too strong, I have to kill you. And it's going to release its perforin, granulysin, and granzymes at the target cell and induce apoptosis. Okay, so that is how natural killer cell activation works. Now it turns out that natural killer cells actually recognize sort of a ratio of inhibitory to activating stimuli on the surface of targets. But that's sort of outside of our scope for you to understand. Another way that they can kill, which wasn't mentioned in the other slide, is this protein called TRAIL, which it has a really weird long name. It stands for TNF-related apoptosis-inducing ligand. So TRAIL is a protein that can, by itself, cause a target cell to die. The way that TRAIL works is it can only tell a cell to die if the cell is expressing a death receptor, usually DR4 or DR5, either of those two. And death receptors are proteins that become upregulated on cells that sort of want to die. For example, cancer cells that have been treated with chemotherapy, like etoposide, and cells that are sort of infected beyond saving. They may express the death receptor on their surface. And natural killer cells are going to swing by, and their TRAIL protein is going to bind to the death receptor, and that causes the trimerization of the death receptors and the subsequent induction of the apoptotic pathway. So the cell undergoes apoptosis if it has a death receptor on its surface that binds the TRAIL. Okay, not a super important thing to know, but this is one other way that natural killer cells can tell a cell to die. And lastly, we have ADCC, which is a topic that I've danced around a little bit too much so far. ADCC is something that a lot of cells can do, but natural killer cells are one of the angriest and meanest cells that do it, and they're really good at it. So here we have an antibody that's binding to antigens on the surface of a target cell. This may be a cell with viral antigens on its surface. For example, a cell infected with the influenza virus is literally putting influenza proteins on its surface. That cell ain't doing too hot already. It's infected with the virus, and antibodies are being made and they're depositing on its surface. Well, a natural killer cell swings by and it says, hey, you've got antibodies here. I'm going to bind to those antibodies that are deposited on your surface, and I'm going to be triggered to degranulate, release my perforin granulysin and granzymes, and you're going to die. So that's exactly what it does. It binds to the IgG molecules on the surface of the target cell, and that triggers the natural killer cell to release its granules that contain deadly proteins. Those proteins kill the target cell. That is antibody-dependent cellular cytotoxicity, or cell-mediated cytotoxicity, whichever you prefer. One of the more important receptors that they use to do this is Fc gamma R3, or CD16. CD16 is just a receptor for IgG. It binds to IgG that has deposited on something, and it triggers the natural killer cell to degranulate when that happens. Now, a number of other cells also express CD16, and those include eosinophils, macrophages, neutrophils, and mast cells. And you may be wondering, does that trigger the same response in all of those cell types when they recognize that there's IgG deposited on a cell? The answer is no. These other cell types do not respond in the same way to the natural killer cell. Eosinophils, for example, they're going to degranulate, but the things that they release are not probably going to be so toxic to the target cell. They're not going to directly induce apoptosis. And macrophages, it may trigger uptake. And neutrophils, it may trigger uptake of whatever the antibodies are deposited on. Mast cells are going to release histamine in response to this. Okay, so different cell types have different responses to the same stimuli because they're specialized to carry things out in a certain way. Okay. And lastly, with natural killer cells, the very important feedback loop between the two cell types, natural killer cells and macrophages. Natural killer cells, they release interferon gamma when they're activated. Interferon gamma activates macrophages. Activated macrophages make IL-12 and tumor necrosis factor, both of which act on natural killer cells and cause them to make more interferon gamma, and it just goes in a positive feedback loop of continual activation. Excuse me. And this is sort of what natural killer cells look like. This is what they look like in the blood. They sort of give off a radioactive vibe because they're just looking for trouble. They have these little pink spots in their cytoplasm. They look kind of like lymphocytes because they technically are a lymphocyte, but this is under electron microscope here, transmission. And to elaborate further on natural killer cells being an innate lymphoid cell. What is an innate lymphoid cell? So innate lymphoid cells, or ILCs, these are lymphocytes that amplify signals delivered by innate recognition, for the most part. Natural killer cells you could think of as being an exception to that. So for most of the ILCs, the cells are stimulated by the cytokines being made by other innate cells during the response. So they're going to be responding to things like IL-12, IL-23, all of this stuff that's being made by immune cells, innate immune cells, okay? And in response to those cytokines, they're going to make more cytokines of their own. And the cytokines that they make can have a really profound impact on the nature of the immune response and the polarization of the immune response. So you have 4 major classes of innate lymphoid cells. You've got the natural killer cells. Each of these subsets is very similar in structure and function to a certain type of T cell, which we'll talk about later in terms of T cells. Natural killer cells are pretty similar in how they go about things to the cytotoxic T lymphocytes, which are your CD8 positive T cells. And they're going to be killing things, but they're also going to be making interferon gamma. Your ILC1s are similar to Th1 cells, and they're going to be promoting cell-mediated immunity by making interferon gamma. And they respond to IL-12 and IL-18 made by activated macrophages and dendritic cells. And the pattern sort of continues with each of these other cells. They mirror a different T cell subset in terms of what they do and what they promote. And a couple of other fun facts. In response to IL-1, your ILC3 cells, they can actually express MHC2, they can produce B7, and they make cytokines, meaning ILC3s are technically a type of antigen-presenting cell as long as they've already been stimulated with IL-1. They can present antigen, weirdly enough. I don't think we've figured out the significance of that yet, but they can express MHC2. That's actually all I want to really say about this right now. And here's all the cells that you can find in the blood just to keep track of them in one place. Your lymphocytes here, these include B cells, T cells, natural killer cells, and the other Cs. Okay. And our last innate cell, our very last innate immune cell, the dendritic cell. Dendritic cells are really important for being antigen-presenting cells. That's their major job. They are antigen-presenting cells. They are important for mounting the adaptive immune response because they present tons of antigen to naive T cells. And we'll talk about what naive means in a second. They are mononucleated phagocytes, so they're in the same sort of subset of cells as the monocytes and macrophages. They're very closely related to these two cell types. Immature dendritic cells are dendritic cells that have not encountered anything that makes them angry. Okay? They have not encountered any bacteria. They have not been stimulated with any kind of pro-inflammatory cytokine, and they're just vibing. They're maintaining homeostasis, and they're kind of doing their thing. They have no reason to be upset or alert attention. Immature dendritic cells, their major job is to engage in macropinocytosis. So they just hang out in the tissues, and they take up a bunch of fluids and solutes, and they break them down into little tiny pieces and spit them back out. So they help to clear tissue of debris, just like macrophages do. However, when they get pissed off, when they've been stimulated with cytokines or they've encountered some kind of pathogen, they become mature dendritic cells. And these are the most important cell type in the entire immune system, probably, at least in the innate immune system, because they activate naive T cells, and they are the only cell that activates naive T cells. A naive T cell is a T cell that has not yet encountered antigen. Okay? So T cells are made in the thymus, and then they sort of float around, they circulate in the blood, and then they enter a lymph node. And in the lymph node, they wait for an antigen to swim by, sort of. Well, they technically wait for an antigen to be presented to them. Okay? Until that point, they're kind of just stagnant. They don't do anything. However, a dendritic cell can take up pieces of a pathogen or a whole pathogen, shred those pieces into tiny pieces, load them onto MHC2, and present them to those naive T cells that are waiting to encounter an antigen. Okay? And in response to being presented with an antigen, a T cell is going to turn into an effector T cell that produces cytokines or can really efficiently kill cells. So T cells are really what's going to whip the immune system into shape and fight off the infection as best as possible. Okay, so mature dendritic cells are very superb also at delivering the co-stimulatory signals and the cytokines that are necessary for activating and causing the differentiation of T cells. They express really, really, really high levels of B7 when they're mature. So they can give plenty of co-stimulation to any T cell that needs it. That makes them really good at activating naive T cells because naive T cells need tons of co-stimulation in order to become activated. They also make cytokines, which are technically another important secondary signal. Cytokines are going to not only tell the T cell that it's okay for them to become activated, but also it's going to tell the T cell what kind of effector T cell it should become. We'll talk more about that in a moment. So the dendritic cell producing cytokines, those cytokines play a very pivotal role in driving the differentiation of naive T helper cells, which we also call Th0 cells. Originally, it was postulated that we had two major kinds of T helper cells. We had the Th1 cells, and then we had the Th2 cells. And we thought that that was the end of the story for a while. We made these big discoveries about how we can make a naive T cell turn into a Th1 versus a Th2 cell. We thought it was all over. Okay? And it turns out that some of the dendritic cell cytokines are going to be dictating what choice of effector T helper cell a naive T helper cell is going to turn into. Later on, it was discovered that there are tons and tons of other T helper cells out there, and each of them has different different stimuli that induce differentiation from naive T cells into that subset. Dendritic cell cytokines also modulate inflammation and local cell behavior, but for the most part, the significance of their cytokines is that they tell T helper cells, what kind of T helper cell to turn into, effector T helper cell to turn into. And basically, they make a lot of the same cytokines as macrophages. The difference is they're making those cytokines for the benefit of the naive T helper cells and also the naive cytotoxic T cells. So we'll talk about those T cells in a second. Dendritic cells are present both in lymphoid tissues and non-lymphoid tissues, and they can also be produced from monocytes that have entered the tissue from the blood. And monocytes can differentiate into dendritic cells. Dendritic cells are the only antigen-presenting cells that are capable of cross-presentation. This is when a cell takes up a peptide from the foreign environment, from the external environment, and load those peptides into MHC1 instead of MHC2. This gives them the unique ability to activate naive CD8 positive T cells without needing to be infected themselves. They are the only cell that can do this. Okay, so let's look at the story of a dendritic cell. It'll make a lot more sense when you see this. So normally you've got these immature dendritic cells hanging out in the tissue, right? However, they then encounter bacteria or they are stimulated with cytokines and they begin taking things up and loading things into MHC2 and into MHC1. They become mature dendritic cells. Mature dendritic cells have a very strong preference to navigate into lymph nodes that drain whatever the tissue is that's infected. Okay, so they're going to enter the lymphatics where they eventually reach a site in the lymph node where T cells are present. All right. And at this time, they've got plenty of peptides loaded into MHC1 and MHC2 molecules on their surface. They just need to find the T cell that's specific for each of those molecules. Okay, so they're going to enter the lymph node from the tissue, and in the lymph node, they're going to sort of browse the selection of T cells that are present there. And they're hoping to find a T cell that they can activate. Okay, so the naive T cells encounter their antigen on the surface of the dendritic cell, and that causes them to become activated. And then they differentiate into effector T cells that go and take care of the problem. Okay. So mature dendritic cells are efficient antigen presenters that deliver a lot of co-stimulation, and they also move to the appropriate place to get you a naive T cell activation. They're really the only cell type that does all of this. Okay, macrophages don't do this. So some things that can drive dendritic cell maturation. Interferon gamma, tumor necrosis factor, type 1 interferons, PRRs as well. So all of your innate immune cells are expressing your pattern recognition receptors. Some of those include the toll-like receptors, which I'll discuss later. All of these things can drive dendritic cell maturation, cause them to migrate to lymph nodes, present antigen, upregulate B7, deliver tons of co-stimulation. They begin producing a bunch of cytokines that cause T cell differentiation. They activate your naive T cells. And it was found also that mature dendritic cells take up larger things than naive dendritic cells. So this is consistent with the idea that dendritic cells normally are clearing tissue of debris, little fragments of debris, whereas when they're angry and mad, they begin taking up pathogens and presenting the remnants of pathogens, the shredded remnants of pathogens to T cells. Okay. So T cell activation looks a little bit like this right here. You've got a dendritic cell on the top and you've got a T cell on the bottom. Signal 1 is the specific peptide loaded into MHC. Okay, and we're looking at the activation of a CD4 positive T cell here. CD4 positive T cells have a T cell receptor that recognizes a peptide that has been loaded into MHC2. They also express CD4, which binds to the outside of MHC2 during antigen presentation. And then you also have a co-stimulatory signal delivered to the T cell by the dendritic cell. B7 is that co-stimulatory signal. B7 is expressed in very high levels on dendritic cell surfaces, and it binds to a receptor called CD28 on the T cell. So the T cell receptor binding to antigen is the activation signal, but without B7 delivering a signal through CD28 to the T cell, the cell actually will not survive the activation signal, because B7 is only going to be present if there is an infection. Something bad is happening if B7 is present. So B7 is like a secondary check to make sure that it's okay for the T cell to actually truly become activated. Okay. And lastly, the dendritic cell is going to provide the naive T cell with cytokines that induce it to differentiate into a certain kind of T helper cell. And we'll talk about specific cytokines that do specific things later on. And as you can see, dendritic cells form the bridge between innate and adaptive immune responses, right? They are the antigen-presenting cells that induce T cells to become activated. There's a lot of different routes of antigen processing and presentation by dendritic cells. If it takes something up through a receptor, it's going to load it into MHC2. And it can technically also load it into MHC1 through cross-presentation. It's the only cell that can take something up and then load it into MHC1. Every other cell type can only load peptides that were generated inside the cell onto MHC1. So it's an important distinction. And then you also have macropinocytosis, which is what they do when they're not so angry for the most part. But that can deliver antigens into MHC2, and T cells can recognize that, CD4 T cells. If a dendritic cell is infected by a virus, those peptides from that virus are going to end up in MHC1, where they're going to get you a CD8 T cell binding to it. And then if the virus was taken up from the external environment, they can do cross-presentation, just like I said. They're the only cell that can load exogenous things into MHC1. Okay. So finally, the long-awaited explanation of what T cells are all about. So the first kind of T cell, there's two kinds of T cells. There's the CD8 positives and then the CD4 positives. Okay. The CD4 positives are the T helper cells, and they express CD4, they express a T cell receptor, and they express CD28. Those are three important things that they express. For the most part, T helper cells are residing in lymph nodes, but they can also circulate, especially once they've become activated and have encountered antigen. Okay? So the T cell receptor on a CD4 positive T cell, it binds to an antigenic peptide, a peptide that has been loaded into MHC2. But part of the T cell receptor recognizes the peptide, another part of the T cell receptor recognizes MHC2. So it's a little bit responsive to the actual antigen presenting molecule. So it recognizes MHC2, and it recognizes that peptide plus MHC2 with very high affinity, extremely high affinity. Every T cell receptor is unique. A single T cell expresses only one T cell receptor of one kind and one specificity, one amino acid sequence. Every T cell has a different T cell receptor that is responsive to a different antigen, a different peptide loaded into MHC2. Okay, so when the T cell receptor recognizes its very specific peptide, that will allow the T cell to become activated in response to that. So these are the cells that dendritic cells present antigen to. These are one of the T cells that they do that to. CD4 is a molecule that binds to the outside of MHC2 during this recognition to stabilize the connection between the T cell receptor and the MHC2 molecule. So I have a picture of that somewhere, hopefully. It's probably in the next slideshow. That's okay. So CD4 is also involved in the TCR MHC2 connection, and it's just going to help hold them together. Okay. And then CD28, which is a receptor for B7, and it delivers co-stimulation to the T cell. Hopefully B7 is present, and if it is, it will bind to CD28, and that delivers a co-stimulatory signal to the T cell, saying, okay, it's fine. There is a problem here. You can become activated. Now, naive T helper cells are sometimes called Th0 cells. These are cells that have never encountered antigen before. It makes them naive. Okay, they've never found their antigen before. They are activated by dendritic cells presenting antigen inside MHC2. Dendritic cells only can activate naive T cells. Cytokines made by the dendritic cell, or maybe other cells in the very small microenvironments surrounding the T cell during antigen presentation, determine which type of effector T helper cell that the naive T cell is going to turn into. And there's a couple of different ones that we're going to learn about. There's the Th1 cells, Th2 cells, T regulatory cells, Th17, and follicular helper T cells. Different signals are going to tell the naive T cell which of these it needs to turn into. Different cytokines are going to specify which of these is necessary. Now, the reason that this is so important, and probably also the reason that it's so confusing, is that there are so many things out there that kill you in so many different ways and hide out in so many different places in your body that we need a lot of different, very super specialized T cell subsets that recognize each of those different things. And they're really good at recognizing each of those different things. Otherwise, we wouldn't get a very good response and it wouldn't be effective, right? So here's a little bit on that. So different effector T helper cells promote different modules of defense because each of these different T helper cells produces different lymphokines upon activation, okay? And so your Th1 cells make different cytokines than your Th2 cells. And those cytokines are better at killing viruses. They're better at promoting antiviral immunity and intracellular pathogen defense. Th2 cells, they're really good at promoting parasite defense. Okay, so they're going to make different cytokines than Th1 cells. It wouldn't make sense if there wasn't this polarization. And each of these has a bit of a different role that they play. And T helper cells can only respond to peptides loaded into MHC2. Therefore, they cannot provide help in response to carbohydrates or polysaccharides or lipids or anything like that. So when a pathogen has a very thick polysaccharide covering, this can inhibit the ability of the host to get a T cell response. So T helper cells are the central players in the immune system. They produce the cytokines that direct the adaptive immune response. However, the initial activation of all naive T cells requires co-stimulation from the dendritic cell that is activating it by presenting antigen to it. Okay, so here's how the differentiation process sort of works. The naive CD4 positive T cell hangs out in the lymph nodes, and a dendritic cell is drained from an infected tissue into the lymph node, carrying antigens on its surface, carrying MHC2 plus peptides on its surface. So dendritic cell presents those peptides in the context of MHC2 and delivers a co-stimulatory signal to the naive CD4 positive T cell. What happens next is proliferation. You get 1,000-fold increase in how many of this cell there were, and then also differentiation, which kind of occurs at the same time, but mostly toward the end of the proliferation period. They're going to turn into, first of all, short-lived effector CD4 positive T cells. These are your T helper cells. Those T helper cells, they originally are going to be in the lymph node, right? Because that's where your naive T cell was, and that's where all the proliferation happened. But as soon as possible, these T helper cells are going to leave the lymph node, circulate in the blood, and eventually they're going to find the inflamed tissue where they're going to look for their antigen again. Usually where they're going to find their antigen is they're going to find it being presented by macrophages that need help. And those T cells are going to say, hey, macrophage, I was activated back in the lymph node by a dendritic cell, and I know that this antigen is associated with a problem. Therefore, I see that it's on your surface, so you probably need a lot of help right now to try to fight this infection off. And that T cell is going to respond to that MHC2 on that macrophage in the tissue by giving it just a ton of cytokines that activate it. Okay. The same thing goes for other cell types. However, there's a little bit of a difference in terms of what cytokines are made by different effector T helper cells. Okay, so your Th1 cells, they're going to make cytokines that help against a virus response. Th2 cells are going to be making cytokines that help against a parasite response. Also, you get differentiation into memory T cells, memory CD4 positive T cells. And it turns out that these memory cells are actually polarized identically to the short-lived effector cells. So you have memory Th1 cells, you have memory Th2 cells, memory Th17 cells. And those memory cells are really long-lived, but they're not really actively looking for a problem. They kind of just hang out in the lymph node until an antigen comes again. They do go to the tissues though, but they're usually just waiting for the pathogen to be reintroduced, right? And then they're going to provide a rapid response to that pathogen if they encounter it. And then you've got your follicular helper T cells, and these are going to be made anytime you get the activation of a CD4 positive T cell. You're going to make some of these kinds of T cells. The job of these is to provide B cell help, and we'll talk more about what these cells later. Okay. We got 6 minutes. I don't know. Do you guys want to call it early today? There's kind of a lot of. Yeah, I bet you are.